This week's lab worksheet reviews the different types of heredity we see and how alleles can combine differently with the union of sperm and egg to create a variety of possible zygotes or children. So we do Punnett Square analysis to predict what the probability of a specific allele combination is going to be. So if we look at our first example on the worksheet for this week, it says it follows simple dominant recessive inheritance. And the disease we're looking at is phenylketonuria, or PKU. So the letter for the allele for PKU is P. So capital P tells us that it is a normal allele, which means that a child who has the big P is healthy, does not have PKU, and little p, which we'll use an underline so we can tell the difference between large p and little p, that is the abnormal allele, and that means if they carry that allele and there is no dominant allele to override it, that child will have PKU. So if we have a person who is big P, they are normal and they do not have PKU. If I have a person who inherited the big P from one parent and inherited little p from the other parent when they were formed at fertilization, because this follows a simple dominant recessive example, that means that the big P here overrides the little p. As a result, they are normal, but they are a carrier. So they're healthy, but they are a carrier of that bad allele, that small p. And then if I have an individual that is little p, so two lowercase p's, that when we look at simple dominant recessive inheritance, that means they have nothing to override that recessive allele, that little p. So that means they are going to have the disorder PKU. So that is a disease state, and that is not an ideal scenario, obviously. So they're unable to break down the amino acid phenylalanine. So when we're starting out looking at these different Punnett Square examples in our lab worksheet this week, we have to know what each letter represents. And there's a separate handout that explains the different letters and what they represent and the type of inheritance. So this first one, like I said, is simple dominant recessive, where the dominant allele is the capital letter, and it overrides the smaller letter. So if an individual is heterozygous, like in this case, one of each, one big P, one little p, they are still normal because the large P overrides the negative or disease characteristics of the little p, but they are a carrier in the sense that they're going to carry that allele. So if we look at an individual, no matter what his genotype is, the genotype is the combination of alleles. So let's say you have a parent that looks like this, like our first example. They are heterozygous.
Hey, Mom.
So when this individual, this heterozygous individual, makes sperm and egg by meiosis, remember meiosis is what happens in the ovaries and testes. In meiosis 1, the alleles are going to separate. And we're going to have one cell getting big P, and the other cell is going to get little p. Then each of these cells is going to undergo meiosis 2, and those alleles are going to separate. And we're going to form two cells that are big P and two cells that are little p. So at the end of meiosis 1, and again meiosis 2, we have sperm and egg that are going to be 50% are going to be big P, and 50% are going to carry the little p allele. And we can just think of this by looking at this zygote here that individual started out with. All of their body cells are going to be big P, little p. But remember, sperm and egg are only N, or haploid, so they have half the number of chromosomes, so they're going to have one allele or the other. So it's just like a coin has heads or tails. This has big P or little p. So any heterozygous individual is going to have the possibility of having 50% of those sperm carrying the big P and 50% of those sperm carrying the little p. So starting out then, looking at a Punnett square, draw this nice box here. We're going to put the parents, their possible sperm and egg possibilities, along the outside of the square. So in example number 1, it tells us that we have big P little p times big P little p. That means that the mother is big P little p, the father is big P little p. So when they make eggs and sperm during meiosis, 50% of their sperm or egg are going to be big P, so that's mother, we'll put her on the side, and 50% of her eggs are going to be little p, so we'll put that down on the side there. Same thing with dad, big P, made this a little too tall. So he's going to be big P, and half of his sperm are going to be little p. So we'll kind of just go like this, make them look like sperm, and these are eggs. So if this sperm and this egg come together, we're going to have a zygote with big P, big P. If this sperm fertilizes this egg, we're going to have big P, little p. If this sperm fertilizes this egg, we're going to have big P, little p zygote. And if this sperm fertilizes this egg, we're going to have little p, little p. So in analyzing your genotypes, on the right side of your example for number 1 on your worksheet, it tells you to list the genotypes. So the genotypes in this example, we're going to have, looking at this, we're going to have 25% are going to be big P, big P. And 50%, 2 out of 4, are going to be big P, little p. And 25% are going to be little p, little p. I'll put an underline there to show that that's a smaller or lowercase p. So if I look at this example, those are the genotypes. That just tells me what are the different allele combinations that I could end up with parents in the different combinations of sperm and egg coming together to make the zygotes, which are inside the Punnett square. So now I can look at the phenotypes, or the physical characteristics. Recall that we said that if any individual is big P, they are normal and they do not have PKU. If they're big P, little p, they're normal also, but they're carriers. So if I look at our example here, I have 25% big P, little p, and 50% big P, little p. So my phenotypes in this example are, I'm trying to get to the next page here. We're stuck, okay. So I'm going to have 75% no PKU. They're going to be normal individuals. And 25%, which is this here, are going to have PKU. So that's my phenotype, is what physical characteristics do we see as a result of the genotype. And that's our introduction to example number 1. I'd like you to try to look at the second example and see if you can answer that one, and then I will follow up with another video. If we look at example number 1 at the bottom, it asks how many phenotypes are possible. There's 2 phenotypes possible. There's either having PKU or not having PKU. And the next question asks how many genotypes are available. Well, for that, I just look inside my Punnett square. Here's one genotype combination, 2 big P's. Here's another genotype combination, big P, little p. And here's the other genotype combination, which is little p, little p. So there's 3 genotypes possible. And that concludes example number 1. All right, now we're going to look at the second example under number 1, dealing with PKU. And it says in the directions for the second example that we have a homozygous dominant parent and a heterozygous parent. So homozygous dominant means that we have a parent that is big P, big P. And heterozygous means we have a parent that is big P, little p. So when we draw a Punnett square for this combination, for this parent, half of the sperm are going to be big P, and the other half of the sperm are also going to be big P, because those are the two alleles that are present in that individual. For the other parent, let's say it's the mother, we're going to have half the eggs will be big P, and the other half of the eggs are going to be little p. So when we go across these individuals, when this sperm fertilizes, this sperm fertilizes this egg, we're going to have a big P individual. If this sperm fertilizes this egg, we're going to have a big P, little p individual. And if this sperm fertilizes that egg, we're going to have another big P individual. And if this sperm fertilizes this egg, we're going to have a big P, little p individual. So these are our zygotes then, the 4 zygote probable combinations based on parents that are homozygous dominant and heterozygous. So the genotypes we have here then, we can see that we've got 50% that are going to be big P, little p, and we have 50% that are going to be big P, big P. So the phenotypes, because all individuals here have the big P allele, which is going to cancel out the negative effects of the little p, we can say the phenotypes is 100% are normal. No PKU, because none of them have the double small p in their genotype. So the phenotype here is all normal. So the question underneath that Punnett square in the worksheet says, what is the probability that a child will develop PKU? The probability is 0%, because none of them are little p, little p. None of the offspring can potentially be little p, little p. So that's example number 2. Example number 3 on page 1 talks about a heterozygous father. So we have big P, little p for the father, and we have a mother with PKU. So that means the mother, in order to have the disease, she has to have 2 recessive alleles for PKU. So that means she is little p, little p. So when we go to do our Punnett square here, again, 50% of the father will have big P, 50% of the father's sperm will have little p, and for the mother, 100% of her eggs are going to have little p, because those are the only alleles that she has. So when we cross these, we have big P, little p. We cross this one and this one, we get big P, little p. When we cross this one and this one, we end up with little p, little p. We cross this one and this one, we get another little p, little p. So the genotypes in this arrangement, the genotypes, we have 50% that are going to be little p, and we have 50% that are going to be heterozygous, big P, little p. The phenotypes, the physical expression of these traits, is that we're going to have 50% that are going to be the little p, so these individuals are going to have PKU. So 50% PKU disease, and 50% are going to be normal. But the question underneath that asks us, what is the probability that a child will be a genetic carrier? Well, these individuals, big P, little p, are heterozygous. Even though they're normal, they are carriers. So the answer is 50% are carriers. Because those heterozygous individuals, even though they're normal because they have the big P that overrides the effects of the little p, they are carrying that in their cells. So when they go to make sperm and egg, they could potentially pass on a sperm or egg with that little p. All right, our second page of the lab worksheet on genetics talks about sickle cell disease. Sickle cell disease is described as an incomplete dominance inheritance example. So if we have sickle cell alleles, the healthy allele is written as HbA. And HB stands for hemoglobin, which means hemoglobin is functional, and when low oxygen environments occur, the hemoglobin can effectively transport oxygen to the tissues. But the bad allele for hemoglobin, it's a recessive allele, it's called the HbS allele, means that hemoglobin becomes dysfunctional in low oxygen environments, like if a person is suffering from asthma or advanced COPD or pneumonia or intense exercise or seizures or high altitudes, anything that causes low oxygen. So that red blood cell, instead of being normally round and plump and able to transport oxygen efficiently, if the oxygen environments get low, the red blood cells become sickle-shaped. So they look like that. And as a result, they can't travel through the capillaries efficiently, and they cause a lot of clotting in the smallest of capillaries, and that impairs blood flow and can affect those tissues that are downstream from that clot that can't get proper oxygenation. So it's a very serious condition. It can cause death. But because it is a recessive allele, in order for a person to truly have full-blown sickle cell anemia, they have to have 2 copies of the bad allele. So in a diploid individual, they would have to have 2 copies of that sickle cell anemia allele, the S. But because it demonstrates incomplete dominance, what that means is if a person has both healthy alleles, just like anything, they're going to be healthy and they're not going to pass on that gene. So that's what a healthy person would look like. So this is the unhealthy sickle cell anemia individual if they have both alleles. But something different happens when we have incomplete dominance in the heterozygous individual. If a person is HbA but has an allele for the sickle cell, so they look like that, so they inherited one healthy allele from their mother but one unhealthy allele from the father, or vice versa, they end up with an intermediate form of this disease, which is called sickle cell trait, which means they're going to have some clotting, some formation of bad hemoglobin here, but it's not going to be as severe as someone with sickle cell anemia. So we call it sickle cell trait. It's an intermediate form. They're not completely healthy, but they're not as sick as someone who would have both alleles. So again, we call it sickle cell trait. So let's look at our first example under number 2. It tells us that we have a set of parents that are HbA, so that's one parent, and the other parent is HbS, HbS. And then it asks us, what are the phenotypes? The phenotypes of these parents, we can say this parent is healthy, no issues, because they do not have that bad allele, that HbS allele. But looking at the other parent, they have 2 bad alleles, so there's 2 S's here, HbS and HbS, so therefore this parent has sickle cell anemia. So that's the phenotypes of the parents. So let's do a Punnett Square analysis here. So looking at this parent, let's say this is the father, half the sperm will have the big A, and the other half of the sperm will also have big A, because those are the only 2 alleles that parent has. The same is true with the other parent. They're going to have HbS and HbS because those are the only alleles that parent has. So when these 2 come together, we're going to have HbA, HbS. Same thing here. All the children, if we do our Punnett Square crossing over like we did in the other examples, they're all going to be heterozygous. HbA, HbS. So therefore, looking at the genotypes of the individuals, 100% are going to be heterozygous for their genotype. And if I look at their phenotype, 100% are going to have that intermediate form, which we call sickle cell trait. So that is example number 1. So let's look at our second example. We have a parent that is HBA, and the other parent is HBA and HBS. So if we look at the phenotypes then of these parents, we can say that this parent is normal, no disease, normal hemoglobin. And this parent is going to have that intermediate form because it is heterozygous. It's going to have sickle cell trait. So as a result of that, we can make a Punnett Square. So this parent will put on top HBA. HBA are the possible alleles that would go to sperm, and egg possibilities would be 50% HBA for this individual, and 50% would be HBS. So when I do my crossing here then, these two would be I'm running out of room here, I need to write a little smaller, HBA. I'm going to have to write this bigger because this is not going to work. Okay, so if I cross these two, I'm going to have HBA, HBA. If I cross this one and this one, I'm going to have HBA, HBS. And the rule is we always put whatever letter occurs in the alphabet, we put that down first. So that's why I'm putting the A first in each example. And then if this sperm and this egg come together, I have HBA, HBA, and I have HBA and HBS. So the genotypes here, I'm going to have 50% HBA, HBA. 50% are going to be HBA, HBS. So my phenotypes then, 50% healthy, and 50% are going to have the sickle cell trait. So they're going to have that intermediate form of the disease. So the question on the bottom, it says, what is the probability of the disease allele appearing in the offspring? Well, the probability, I can see one here and I can see one here, so that's 2 out of 4. The answer would be 50% is the probability of having the disease allele in the offspring. And true, false. The offspring with the disease allele are genetic carriers in the strict definition of the term. The answer to that would be false, because carriers are unaffected people that have the disease allele, so they're unaffected. But again, remember, in incomplete dominance, they are affected. They have this sickle cell trait if they're heterozygous. So the answer to that is false. They are not considered genetic carriers because they display the disease in a lesser form, but they still are affected by having that little s. So let's look at our last example on page 2 here. It says both parents have minor problems. So when we say minor problems, that means it's telling us they have sickle cell trait. So the genotypes of these parents are both going to be HBA and HBS. So that's one parent, and the other parent is the same thing, HBA, HBS. So when I do a Punnett Square of these individuals, this parent will be HBA, the other 50% of sperm will be HBS, same thing with the mother, 50% of eggs will carry the HBA, 50% of eggs will carry HBS. So if these two come together, we have HBA, HBA. These two come together, we have HBA, HBS. These two come together, we have HBA, HBS, and these two coming together we have HBS, HBS. So the questions here, now the genotypes, we're going to have 25% are going to be HBA. So these zygotes or children will get out lucky in the sense that they will not inherit that disease allele from either parent, and they'll be completely healthy, and that allele will be gone in that genetic line. So 50% are going to be heterozygous, so 50% will be HBA, HBS, and 25% here are going to be HBS, HBS. So the same follows through with the phenotypes then. If I look at the phenotypes, I can say that 25% are going to be normal hemoglobin. So these individuals are the normal ones. Then we're going to have 50% are going to have the sickle cell, I'm going to abbreviate just to have room here, sickle cell trait. So that's these individuals that are heterozygous are going to have that intermediate form of the disease. And 25% are going to have sickle cell anemia, which is the serious form of the disease because they have both of those recessive alleles. So those are the examples on page 2 dealing with sickle cell anemia. Right now we're going to look at example number 3 from the lab worksheet on genetics. And this one talks about blood types. And blood types is an example of another type of inheritance which we call codominance. And codominance means that there are two alleles that neither one is dominant over the other. And in the case of blood type, we use the capital letter I, and the allele is A. That's the one dominant allele, and the other dominant allele is B. So we have two alleles that are dominant, so A and B. So if a sperm contains B and it fertilizes an egg that contains A, the resulting zygote is going to be IA, IB. So one is not dominant or the other. So that person is going to be blood type AB. So we always have two alleles. So A and B are possible alleles. But if the other allele is neither A nor B, there is a recessive type allele. And to indicate that, we use the lowercase I. So possible combinations for blood type might be IA, little I, or IA, IA. Whoops. And in both situations, the blood type of these individuals would be blood type A. So in this situation, we would say that the individual is heterozygous because it contains the lowercase I. And this would be homozygous dominant in that case because they have both chromosomes, one from mom, one from dad, both contained the A allele. So let's look at some other possibilities here then. If a person is blood type B, they can be capital I, B, IB, again homozygous dominant, or they can be IB, little I. And in that case, they are blood type B. Now what happens if a person is type AB? We already looked at that example. They would have to have both alleles, A and B, and that would make them type AB. Now, what would happen if a person inherited the two small recessive alleles, little I, little I? In that case, those individuals are neither A nor B. We call them type O. So that's the combination that would result in a type O individual. So kind of understanding a little bit how these alleles are represented in the phenotypes is important to understanding and being able to do some analysis of parents and inheritance patterns. So let's look at example number 3, our first example there at the top. It gives us 2 parents, and it says that one parent is, I'm going to erase this, it says that one parent is I, B, I, B, and the other parent is I, A, little I. So in this case, they are heterozygous for the A allele. So if we go and look at a Punnett square of these 2 parents, half the alleles for the one parent will be IB, and the other half will also be IB because those are the only alleles that parent has. The other parent is going to have IA and little I. So when we combine these, we always put the letter that's first in the alphabet first. So the zygote resulting from this potential fertilization would be IAIB. Looking at this combination, sperm and egg, we would get IB little I. This combination, this combination here would be IA, IB, and lastly, this combination here would be IB, little I. So the question is, what are the genotypes? We're going to have 50% are going to be IAIB, and 50% are going to be IB, little I. So as a result, the phenotypes, we're going to have 50% are going to be type AB blood, because again, they're codominant. One does not override the other. And 50% are going to be type B, because the little I is considered a recessive allele, so this individual is going to have whatever the dominant allele is, either A or B, and in this case it's B, so it's type B blood. Now the question is, what is the probability that a child will have a heterozygous phenotype? Well, the heterozygous phenotype in this example is going to be this individual here, because in a situation of codominance, this individual in the green is not considered heterozygous. Heterozygous means there's one recessive allele, and in this case there is no recessive allele for the green. They're both codominant. So 50% would be heterozygous. And that's the IB little I individual. And then what is the probability that a child will have a codominant genotype? Well, that's what I circled in green there. So the answer to that one is also 50%. Is going to be IAIB, which is considered codominant. They're going to be type AB. Our next example has a parent that is IBIB, and the other parent is little I, little I. So in our Punnett square analysis, the 2 alleles for the one parent are both going to be the same. They're going to be IBIB, and down the side of the other parent, little I and little I. So when those 2 come together, we're going to have IB little I, IB little I, IB little I, and IB little I. So the phenotype, remember, of this parent here, the phenotype of that individual is going to be type B, and the phenotype of this parent is going to be type O, because they have both recessive alleles. So the genotypes of the offspring then, looking inside the Punnett square, these zygotes resulting from these different sperm and egg probable combinations, the genotype, we're going to have 100% are going to be heterozygous for the B allele. And the phenotype is 100% are going to be type B blood. And the question at the bottom of example 2, under number 3, is what is the probability that a child will have the same genotype as the parent? Well, here's the genotype of all the kids. That doesn't match either parent because this parent has both B alleles. This parent has both little I, so the chance of them having the same genotype in this case is 0%. And the second question, what is the probability that a child will have the same phenotype as one of the parents? Well, the children are 100% type B blood, which matches one of the parents. So what is the probability that it'll have the same phenotype as one of the parents? The chances are 100%. Because type B blood is the same as the one parent that is IB. Our last example on page 3 talks about two parents that are heterozygous for type A blood. So what that looks like is we have a parent that is type IA little I. So both parents are going to be IA little I. So in a Punnett Square analysis then, I'm going to put capital IA there, little I for the other allele for one parent, and then the other parent's alleles will go along the side, and we have IA for this combination, and this combination. I'm going to have IA little I, same as this combination, and then this combination would be little I. So my genotypes then, I'm going to have 25% are going to be IA, 50% are going to be the heterozygous IA little I, and 25% are going to be little I. So my phenotypes then for this combination of alleles, I can say that these two individuals, or these two types, I should say, allele combinations, both contain the dominant A allele, and this is recessive, so I can say that 25% plus 50%, 75% are going to be type A, and 25%, this group here, has the two little i's, so 25% are going to be type O blood. So 25% with the little I are going to be type O blood. So the question at the bottom of that third example on page 3 is, what is the probability that a child will have a different phenotype from the parent? So both parents are type A because they are heterozygous. They are type A, or capital I, A little I, capital A little I. So the probability that they'll have a different phenotype of the parent is this group here, this 25% type O. That's a different phenotype than either parent, which are both type A. So therefore we can say 25% probability that you're going to have a child that is type O. Our last example of a type of inheritance pattern is looking at sex-linked traits. And if you recall from the PowerPoint, sex-linked traits are those traits that are carried on either the X or the Y chromosomes, which are the sex chromosomes. In most cases, the bad alleles we see, the disease alleles that are sex-linked, often are carried on the X chromosome. And the one we're going to look at in example number 4 is hemophilia. Hemophilia A is a clotting disorder, and individuals that inherit the bad allele for this disease have trouble clotting, and when they cut themselves, they are at risk for excessive bleeding. So they need medical intervention, or they need to administer a clotting factor to stop the bleeding. So let's look at how we represent sex-linked traits and alleles. So the allele for normal blood clotting is capital H. And the hemophilia is little h, so it's a recessive allele. But because they are sex-linked, we have to assign what chromosome these alleles are found on. So in our first example, we have a parent that is X big H, Y. Now there's no allele for the Y because this allele H for hemophilia is only carried on the X chromosome. And we know from our PowerPoint that any individual who inherits one X and one Y, they are male. So this is the father in this situation, and the mother is X big H, X little h. Now, when we have two alleles for hemophilia, the dominant and the recessive, again I said this is the recessive allele, this is what would cause hemophilia. But if the dominant H is there, this person is only a carrier. So we would say this individual here who is heterozygous for the hemophilia, the mother in this case, because there's two X chromosomes, we call her a carrier. So she will not have hemophilia because the good H overrides the little h and suppresses that disease. So she is going to be a carrier in this case. But now because the male does not have an X, another X that could possibly override a bad hemophilia allele, he can never be a carrier. Only females can be carriers. So I'm going to write that here. Only females can be carriers. Okay. So let's look at our first example then, where we have these two individuals. We're going to draw a Punnett Square here. And we'll put the father on top. So we have the X, capital H, Y. Those are the two alleles when they separate at meiosis to make sperm. And the possible egg combinations for the female are going to be X, capital H, and X, lowercase h. So when these two come together, we have XH, XH, and they just formed a girl. If these two come together, we have X capital H, X little h, another girl. If these two come together, we always put the X first because it comes first in the alphabet. It's going to be XH, Y. And these two coming together, we're going to have X little h, Y. So just like we would expect, we have 50% of probability being male, 50% probability of being female. So when we look at the genotypes for this sex-linked example, we have to separate out the males and the females. So in this case, the males, in our example, we're going to have, I'm just going to use the symbol for male in science is this. This is the symbol for male. Okay, so the male in this case, we are going to have of the males, so 2 males total, so 1 out of 2, or 50%, are going to be normal hemophilia allele, so no disease, but we're just looking at genotypes right now, and the other 1 out of 2, or 50%, are going to have the hemophilia allele. And no other counterpart to override that. So the females, in looking at them, we can see that we're going to have 50% are going to be homozygous dominant for hemophilia, and the other 50% are going to be carriers. So in that case, I'm going to say that the females, the symbol for female is this in science. So the females, we're going to have 50% are big H, and the other 50% were big H, little h. Again, looking back at the previous screen. And looking at phenotypes for these then, of the males, I'm just going to abbreviate using that symbol, of the males, what can we say? We can say that looking at this example of the males, circle this here, this group here, they have the big H, which is the healthy allele, which means they're going to be healthy. and 50% are going to have hemophilia. So 50% healthy, and 50% are going to have that clotting problem of hemophilia. And of the females, looking back, we can see our female, oops, I had them on the other page. Our females, we can see 50% normal and 50% also normal, although they're carriers of that small allele, they don't express it. So we can say here 100% healthy. So the question at the bottom of that first example on page 4, it says, considering all the offspring, what is the probability that an affected child will be produced of all the offspring? So in looking at all the offspring here, which one of these represents an affected child? Well, here we see a normal allele, normal allele, normal allele. Here's the group that has the small h, which means disease. So we can say that this is the group that's going to be unhealthy. So of all the kids, 1 out of 4, this group here, 1 out of 4, or 25%, are going to be, so 25% of all offspring will have hemophilia. Again, probability, right? This is not actuality, because just because we flip a coin 10 times doesn't mean we're going to get 5 heads, 5 tails, but this is probability. This is just making predictions. The second question at the top of page 4 example, it says, when an affected son is born to normal parents, what is understood about the parent generation? So here we have an affected son. That's what's outlined in blue here. That's an affected son. In order to have an affected son, we know that the dad, in this case, gave the Y to make him a son, so he had nothing to do with that hemophilia allele, that little h. The mother gave it. And again, it says the parents are normal. So if the parents are normal, where did that little h come from? The little h came from the fact that the mother was a carrier. Okay, so she was a carrier. She had this little h allele. So whenever we see a normal parents and an affected son, we know that the mother was a carrier. So she is heterozygous for that hemophilia recessive allele that causes the disease. So she is a carrier. Now the second example on the bottom of page 4 talks about a homozygous recessive and an XHY parent. So X big H, Y, that's dad because of the presence of the Y chromosome, and then a homozygous recessive mother. So that means she is little h, little h. So right away we can say we know that she has hemophilia because she does not have a healthy h to override those two little h's. And we know that dad is normal because he has the normal big H and he doesn't have a little h. Even if he did, it would have to be on the X. So his Y doesn't have any allele for hemophilia, so he's normal. So the parent phenotypes is normal dad, hemophiliac mother. So looking at the combination of alleles here then, for these two parents, we have dad here. Mom will put along the side. So we have a daughter that will be a carrier, another daughter that's going to be a carrier. The sons are out of luck. Both sons are going to be little h, because that's all they can inherit from the mother on that X, is the little h. So we have, looking at the genetics then, we're of the males, we are going to have, looking at this group here, these are the males, 100% are going to be X little h Y. My handwriting is not the best, I apologize. That's not much better. Anyway, X little h Y. And then of the females, we have 100% are going to be carriers with the big H and the little h on those X chromosomes. And then the phenotypes of the males, we can say 100% because they had that little h are going to be having hemophilia. And of the females, 100% are going to be normal. But again, because they had that little h, we're going to also call them carriers. So at the bottom of page 4, it says, what is the probability that a child would inherit the disease allele? So not necessarily the disease itself, but the allele. And in each of these cases, when we look, we can see little h, little h, little h, little h. So the answer to that first question would be 100% chance that they're going to inherit that disease allele, because the mother only has the disease allele in all of her eggs, so there's no possibility of avoiding that allele. What is the probability that a child will develop hemophilia? Well, if we look at all the children here, we would say, we can just circle the boys here, 50% of all the children, 50% will develop hemophilia, because those boys are going to have hemophilia because they only have the little h allele. Our last example on the top of page 5 tells us that we have a hemophiliac father and a heterozygous mother. So a hemophiliac father is going to look like this. He's going to have the big X from the mother, but he's a hemophiliac, so that means he has the little h and the Y, which doesn't carry any allele for hemophilia. And the mother is heterozygous, so we know that she's going to be big H, little h. She's going to be a carrier, but she's going to be normal. So these are the genotypes of the parents. The father is X little h, Y, and the mother is X big H, X little h, heterozygous. So in crossing these two parents, we can put those alleles. We'll put the father here, his 2 sperm possibilities, the mother's 2 different egg possibilities. This sperm and this egg come together, we're going to have a heterozygous female. And these 2 coming together, another heterozygous female. Oops, sorry, not heterozygous. That would be homozygous recessive. Little h, little h. And then these 2 coming together, we're going to have a male, big H, and a little h, male. So looking at our genotypes for the males, we are going to have 50% are going to be big HY, and 50% are going to be little hy. The females, we're going to have 50%, or 1 out of 2. Remember, that's where these percents are coming from, because only 2 are going to be male or female. So 50% are big H, little h, and the other 50% are little h, little h. Of the phenotypes then of this combination, I'm going to put on the next page, but let's just look at the Punnett Square to predict that. So of the females, we can say that 50% of the females are going to be carriers, and 50% of the females are going to have hemophilia, because they're going to have 2 copies of that recessive allele. So of the females, 50% carriers, normal, and 50% are going to be hemophiliacs. Of the males, looking back, of the males we have normal, 50%, because they have the big H, and hemophiliac, because they have the little h. So this, in this case, 50% normal. They are not carriers because they don't carry the little allele like the females do. We can see here the female carries that little allele, but the male, there is no allele there, so they are not carriers. and 50% would be hemophiliac. So looking at the bottom of the sheet, the questions there, not in the bottom but in the middle, it says, what is the probability that a child will develop the disease? So if you want to just look at this Punnett Square, who has the disease here, we can see that this group here has the little h's without a big H counterpart. So we can say 50% of the children, 50% of all kids, we'll just abbreviate instead of offspring, or child of all kids will have, we'll just say will be hemophiliac. They're going to have that clotting disorder. From which parent do affected male offspring inherit the disease allele? From which parent do they inherit that allele? So male offspring. So these males are here. Here's the male offspring. From which parent did they inherit the disease allele? It would have to, and that would be in this case, it would have to be the mother in both cases, right? Because if they're male, they inherited the Y chromosome from dad, and that had nothing to do with hemophiliacs. Or hemophilia. So it has to be from the mother. So the second question there is, from which parent? The mother is the answer, because the X chromosome carries that allele. And in this example, from which parent do carrier female offspring inherit the disease allele? So of those 50% that are carriers, so that would be looking at, let's see, looking at the females, the carriers here, so that would be this here. Who inherited this? Or from which parent did they inherit it? Well, we look at a female, dad has a little h, that's the only one that he can inherit. So he can only pass on the disease allele. If she's a carrier, that means her other allele is big H. So we're focusing on this group right here. So this, I'll just write it down here, this individual is a carrier. So the only way that they can be a carrier means that they had to get the disease allele from dad. Because if they have the big H, the only way they would have a big H and be a carrier is because they got the big H from mom. So that little h had to come from dad. So the third answer to the third question is from the father. So that concludes our lab worksheet on genetics. If you have further questions, don't hesitate to email. We can sit down and go through them together. With a tutor is also able to work through that with you. And the example on the bottom of page 5, I'm not going to work through those. That's just an extension kind of for extra practice, but the answers will be on the answer key that I will provide for the lab worksheet.